Okay, everyone, welcome back to the final lecture in Anatomy and Physiology 2. So congratulations on getting to this point. And we're going to end it with a relatively interesting and cool topic on patterns of inheritance. So on your right here, this is called a karyotype. And it is a picture taken of your 23 pairs of chromosomes. So I'm not going to do it for you, but if you want, you can count it out. That's 23 pairs of chromosomes. And that's not how your chromosomes look in your body or in the nuclei of your cells. This is done through a special technique of taking them and putting them together in their specific pairs of homologous chromosomes and taking a picture of them. But that is what is called a karyotype. Now, there's a couple of background pieces of information that I want to fill you in on so we can have a conversation about patterns of inheritance. Um, each human body cell, like I just told you, has a full complement of DNA stored in 23 pairs of chromosomes. That's 23 pairs, 23 from mom, 23 from dad, 46 total chromosomes. That's what constitutes being a human. Other organisms have more or less of these chromosomes. You get 22 pairs of autosomal chromosomes, or sometimes called the body chromosomes, and one pair of sex chromosomes, the X and the Y if you're a male, or 2X if you're a female. I just told you that arranging them visually, like you saw before, and like we'll see later, is called a karyotype. Homologous chromosomes are those that make up a complementary pair. They have genes for the same characteristics in the same location on the chromosome. So if we just took a pair, let's say pair 5, and I'm just totally making this up right now, but let's just say eye color is on gene 5, the gene for eye color is on chromosome pair 5. And so on the copy from mom and the copy from dad, you have a gene for eye color. Okay, and nowhere else in those 23 pairs will you have eye color genes. You can have 1, 2, even more forms of a gene, and those are called alleles. Like for eye color, think you could have blue, you could have green, you could have brown, you could have hazel, every other color. I don't want to leave anyone out. But the different, we call these alleles, and they are different forms of the same gene. Okay, so they're the gene for eye color, but they're different forms. You could have 2 copies of the same allele. Let's say you had 2 blue copies. Well, you would be called homozygous. Or you could have 2 different alleles, a blue and a brown, or a blue and a green, or a green and a brown. And if you had 2 different alleles, we would call that heterozygous. The alleles that you have, the genes that you have, are your genotype. The way that you look, the physical characteristic that's produced by that genotype is called your phenotype. So genotype determines phenotype, okay? Let's talk about expression of these alleles. The expression of allele can be dominant, and that would mean that the dominant allele will mask the expression of what is called the non-dominant or recessive allele, but sometimes that dominance can be complete, at other times it's incomplete, and in some cases both alleles are dominant, and we call that codominance. Although any one person can only have 2 alleles corresponding to a gene, there can be more than 2 alleles to exist in a population. A good example of that is ABO blood type, where the alleles are IA, IB, and little I, or sometimes I0, because there's ABO, right? And so there's multiple alleles, but you can only have, as an individual, 2, one from mom and one from dad. They can either be the same or they can be different, okay? Here's a good example. Normally, this is called a Punnett square, and this is a way in which you can determine outcomes from what parental alleles they have and what their offspring might have. So let's say that in this case we give capital B to brown because brown is a dominant allele, and blue will be recessive in this example. And so if you have one capital B, at least one, you only need one because it's dominant and it is completely dominant, you will have brown eyes. In order to have blue eyes, you must have 2 little b's, okay? Now, in order to get in order for you to pass on, let's say the mother has blue eyes here, the father has brown eyes here, each one of these will become a sperm or an egg. You can think of it this way, a sperm or an egg. And the father and the mother, or whatever way you want to say it, the parents can only give at one time one of either their big B or the little b, or either this little b or this little b. They can't give both, and they have to give just one at a time. So what you do is basically, if you were to make a Punnett square, you would put the alleles that each parent have on the sides and just cross them over. So we would bring this big B down here, and this little b down here and here, this big B here, and this little b here and here. And what this shows you is the potential percentages of offspring. So if we took two heterozygous individuals, they're heterozygous because they have two different alleles, a big B and a little b, a brown and a blue, and they had children, their offspring would be, one would be homozygous dominant, two would be heterozygous, and a fourth would be homozygous recessive. When you have two littles, you call them homozygous recessive, homozygous dominant. Notice you don't say heterozygous dominant because heterozygous just already means two different, okay? But, so the genotype would be 25% homozygous dominant, 50% heterozygous, and of course 25% homozygous recessive. Now, that's the genotype. The phenotype would be 75% brown and 25% blue. Okay, now let's see what we might get if we cross or have children with a heterozygous male and a homozygous recessive female. You do all your dragging across and down, and what you get is 50% brown and 50% blue, and the genotype is 50% heterozygous and 50% homozygous recessive. Okay, so hopefully you understand how this is working. Now, let me point out before I go any further that this does not mean if you have 4 kids in this situation, if these parents end up having 4 kids, that doesn't mean one kid will be this, one kid will be this, one kid will be this, and one kid will be this. That's not what that means. It is a percentage. When you flip a coin, you have 50% chance of heads and 50% chance of tails every single time. That doesn't mean that if you flip it twice, you'll get heads and tails, right? You could get heads, you could somehow, unlikely, get heads 100 straight times, right? Very unlikely, but it is possible. Basically what it's saying is that the chance that you have every time you produce another zygote, the chance that it will be this is 25, and 25 meaning 50, and this will be 50. But it doesn't mean that if you had 100 kids, they couldn't all have brown eyes or they couldn't all have blue eyes. It's a 50% chance every single time. So I hope that makes sense. I know it's a little confusing if I'm not explaining it correctly, but I think that I am. I think you're going to get it. So let's move on. Let's talk about Gregor Mendel. You might have heard Mendel's theory of inheritance. Before I go any further, I want to tell you that a theory in science is not like a hypothesis. A hypothesis is an educated guess, whereas a science theory is something that is supported by multitudes upon multitudes of support work being done. And so Mendel's theory of inheritance. So Gregor Mendel was an Austrian monk working in the mid-1800s on this theory of inheritance before we knew anything about chromosomes. It wouldn't be until much, much later that we discovered chromosomes. But Gregor Mendel was an Austrian monk, and as an Austrian monk, he didn't have a lot of stuff to do. But what he did have was this garden where he planted things and kept copious amounts of notes. And he discovered that these garden peas that he would grow were able to transmit their physical characteristics to their subsequent generations in a discrete and predictable fashion. So some of the things that he looked at were the height. There were tall plants, there were short plants, there were plants that had purple flowers and white flowers, there were plants that made green peas and yellow peas, there were plants that made shriveled peas and smooth peas, and he determined that each of those characteristics, he would be able to predict what the offspring would look like. When he mated or crossed 2 pure breeding pea plants that differed by a certain characteristic, the first generation offspring all looked like one of the parents. So the classic example here is he took tall plants that would always make, when he put 2 those tall plants together, they would always make tall offspring, and he would take 2 shorts and put them together and they would make short offspring. And so then what he would do is take one tall plant and one short plant and make them have babies. And what would happen is all of the offspring would be tall. And so he claimed that tallness was dominant because it was expressed in offspring when it was present in a purebred parent. He called shortness recessive because it would be masked in the offspring if one of the purebred parents possessed the dominant characteristic. So what I want you to note is that tallness and dwarfism or shortness are variations of one characteristic. They are alleles of one gene, the height gene. And he called those variations a trait. We now know that those traits are the expression of different alleles of the gene encoding height. Then what he did was he would take the second generation offspring of purebred parents with each other, and he found that the recessive trait would reappear in the third generation in a ratio of 3 dominant to 1 recessive. So what he would do is he would take those tall plants that came from the tall and the small, that all produce tall. Then he would take 2 of those children, it's plants, so it's not weird, and combine them, cross them, and they would make a ratio. If you made hundreds upon thousands of these plants, which that's what he did, you would find that 25% of the time their offspring would suddenly be short again. Remember, all of these parents looked tall. Okay? He proposed that characteristics like height were determined by heritable factors that were transmitted one from each parent and inherited in pairs by offspring. So this, again, he's proposing chromosomes here. He does this in the mid-1800s. And by the way, when he went to present this research, he was kind of laughed off and unfortunately passed away before his work was then rediscovered in the mid-1900s. And the scientist that rediscovered it was like, oh my God, he was right all this time. So unfortunately, didn't really get the recognition that he totally deserved. But anyway, needless to say, very impressive work. So here's basically what he did. He took, let's say these were the pure breeding tall plants, and he crossed them with pure breeding small plants. And what he found was that 100% of the offspring are dominant. But this says AA dominant, but this is not the case, because what you actually get are 100% heterozygous. They're heterozygous, but they show the dominant trait. So all of them will be tall. But note that the small trait doesn't disappear. So that when you cross 2 of the heterozygous individuals, you can get 25% of the time homozygous dominant, 50% of the time heterozygous, and 25% of the time the reappearance of the short trait. So what you found here is that the recessive trait is not disappearing, but it is coming back in the third generation. Okay. He also included crosses that would involve multiple traits, like, okay, this is a tall purple with a small white, and a tall smooth pea and a tall crinkly pea, okay? And so basically this prompted him to formulate the principle of what is called independent assortment. The law of independent assortment shows that members of one pair of gene, which are the alleles from a parent, will sort independently from other pairs of genes during the formation of gametes. And what that means is the tall gene and the purple gene don't have to go together. They can go in separate gametes. And this is really, really good. This works really well for pea plants because they have color, height, seed type, and those things all do sort independently of one another. And it holds true in all alleles, except when they end up happening to be really close to each other on the same chromosome, and then they're unable to independently assort. But the process of independent assortment, even in us, provides for a great degree of diversity in offspring. It's the reason why you don't look exactly like your sibling or your cousins or your mom and dad. And you definitely don't look like the stranger that's sitting next to you in class. And so it's just another way in which we get a great degree of genetic diversity. Let's talk about when things are autosomal dominant. So autosomal meaning they're part of the non-sex chromosomes, and it's basically what we just talked about, where the dominant trait always takes over for the recessive. It always will dominate it, so you only need one copy of the allele to present the dominant trait. Here's an example in which you have a dominant individual crossing with a homozygous recessive individual, and just in the case that there is one allele, every time that allele is passed on, you get the trait, and so 50% of the time you will get a whatever this trait is. It could be a disease, it could just be whatever, a regular old trait, and 50% of the time you will not show the disease or the trait. You will be recessive. If it is autosomal recessive, what that means is you need two copies of the trait in order for it to be shown, for the phenotype to show. So the mother and the father could both be have the trait but not be sick. Let's say this is cystic fibrosis. They could be what is called carriers, where their phenotype is normal, but their genotype, they carry the trait for cystic fibrosis. So when they have, every time they have a child, there is a 25% chance that they will both give their small c and make a child that has cystic fibrosis. Okay, and so this is what this results in some of the rarer diseases that we do see, where parents could be absolutely unaffected but produce a child that is affected. But again, they will also produce offspring that are carriers themselves, and then 25% of the time they will produce a son who is unaffected, or a daughter, could be a son or a daughter. Now we'll talk about X-linked dominant or recessive inheritance. An X-linked transmission pattern involves genes that are located on the X chromosome, which is the sex chromosome on the 23rd pair. Recall that if you are a male, you have one X chromosome and one tiny little Y chromosome. And when a father transmits a Y chromosome, the child is always male, and when he transmits an X chromosome, the child is female. What chromosomes on the 23rd pair can the mother transmit? Well, only an X chromosome, because both of them are X. Okay, if the trait is an X-linked dominant trait, the dominant obviously is dominant over the normal allele, and the pattern is described as X-linked dominant. So here we have an affected father because he has an X chromosome that he received either from his mother or his father. No, he has a Y chromosome, so this X affected is from his mother, and we have an unaffected mother. And if they have children, there is a chance, well, there is a very high chance that every daughter they have will be affected, because she only needs one X chromosome to be affected, and that's what she's going to get, because if it's a daughter, she's going to get this X chromosome. Mother is unaffected, so whenever the son is born, if there is a son born, he will be unaffected because the father is giving his Y chromosome. Now, if we have the affected mother, things are a little bit differently, happening a little bit differently, because mother can either give our affected X or our regular X. And so what you see is if father gives a Y, well, it could be an unaffected son, but it also could be an affected son if the X that's given from mother is the affected one. In the same instance, father can give his unaffected X, which will be a daughter, but if mother gives the affected X, it'll be an affected daughter. And if father gives an X, it'll be a daughter, but if she does give her unaffected X, she will be an unaffected daughter. So 50% of the time, we are going to have the offspring affected, whether or not they are sons or daughters, if the mother is the affected. Okay, X-linked recessive, how does this work? It's much more common, actually, because females can be carriers of the disease, yet still have a normal phenotype, right? Because they'll have one normal dominant X-linked, and they can carry but not be affected. Diseases transmitted by X-linked recessive inheritance include color blindness, blood clotting disorder, hemophilia, and some forms of muscular dystrophy. With X-linked recessive diseases, males either have the disease or are normal. They cannot be carriers, right, because they have a Y chromosome. Females can be normal or a carrier who's phenotypically normal or be affected with the disease. But as you can imagine, X-linked recessive disorders are going to be much more effective on males than females. One example is colorblindness, which affects at least 1 in 20 males, but only about 1 in 400 females. The reason for that is you would have to have a colorblind male with either a colorblind female or a colorblind carrier female, and for them to make a daughter, the probability of that is pretty low. So here is what it looks like in an X-linked recessive. We have the unaffected father and a carrier mother, and when she can obviously give her affected X or her unaffected X. If she gives her affected X to the son, well, unfortunately, he's going to be affected. Let's just say this is colorblindness. If she's carrying the colorblindness trait, well, father didn't give an X, so there's no dominant X to mask this. The Y does not mask it. It's an affected son. The only other time that she gives her affected X is to her daughter, but this time, if it's a daughter, the dad, the male, had to have given an X, and that can mask the recessive trait. So she will become a carrier just like her mother. And then if mother gives the unaffected X, both the son and the daughter obviously will be unaffected as well. Here are just some other examples of how dominance can work. Here's incomplete dominance. This is pretty cool. This happens in flowers a lot, where you get the dominant trait and the recessive trait, and they mix. And when you have the heterozygous trait, where you have the dominant trait and the recessive trait, there's the red here in this instance is not completely dominant, so you get a mixing and you get these pink flowers, so that's pretty cool. And then there's something called codominance. Here's an example of incomplete dominance, and here's codominance, where if you got red and white, red being the dominant, white being the recessive, again, where both are shown equally, and that is called codominance. So very interesting as well. Okay, so here I'm going to leave you with this one. This is another karyotype, and let's take a look. There are 23 pairs. Here's 22 autosomal in the 23rd chromosome. The first thing I'll ask you, is this a male or a female? Well, you should say this is a female, right? Because there's no Y chromosome. There's 2 X chromosomes. And does anything else look weird about this karyotype? Well, if you see, all of them look normal except when we get to 21. And look at that, there are 3 copies of 21. And this is called trisomy 21. If you're familiar with what trisomy 21 is, it is Down syndrome. And so Down syndrome is one of the rare, I'll actually say, instances in which a human can be born and survive for an extended amount of time with the wrong number of chromosomes. They obviously do have some deficiencies in life, and their life expectancy is lower than a healthy individual, but they can survive into adulthood, obviously. There are other examples here that I won't get into with different either deletions of different chromosomes, either autosomal or sex, additions of a sex chromosome that I wouldn't get into in this class, but just interesting to think about. Here's a good exam question, for example, like, is this a male or a female? Very simple, but things like that. So that's what to expect, and I will congratulate you on reaching the end of this class. Good luck in your future endeavors and on the final exam.